Hello, everyone. Welcome to GBI Biomanufacturing's webinar on antibody oligo-conjugates. I'm Andrew Madoc, VP of Technical Operations for GBI Biomanufacturing. We'll get into the technical aspects in a moment, but first, a little information about GBI Biomanufacturing. Our mission is to create complex biologics from molecule to market for innovators committed to improving patients' lives. We mitigate the manufacturing risk by being the preeminent provider of high-quality, cost-effective, flexible, timely, cGMP-compliant manufacturing solutions. This includes preclinical development through commercial supply. GBI has been in business for 32 years and worked on over 500 projects with hundreds of satisfied clients. Additionally, for a small CDMO, we have supported 3 BLA PAS filings. Our capabilities run from 5-liter to 2,000-liter bioreactors for drug substance manufacturing. Additionally, we have drug product manufacturing capabilities. Now a little bit about AOC market by the inference from the nucleic acid market and the ADC market. The global nucleic acid therapeutics market was valued at $4.1 billion in 2021 and is projected to reach $12.2 billion by 2031. This is growing at a compound annual growth rate of 11.6% from 2022 to 2031. Market size and trajectory of nucleic acid therapeutics and additional resources pin that the siRNA market greater than $67 billion by the end of 2023, with an annual growth rate of 18.5% between 2024 to 2036. Certainly worthy of attention. Using targeting moieties to deliver these therapeutics, traditionally naked via lipid nanoparticles or other non-specific delivery modalities, is in its infancy with roughly a couple of dozen programs headed to the clinic, with many more in development. The expectation is that the trajectory could approximately parallel or exceed ADCs for cytotoxic payloads around US $11.3 billion in 2023, projected to grow to $27.4 billion by 2031. I'm going to turn the presentation over to Dr. Mukhtar Sisay, our CSO and VP of Bioconjugation.
GBI has been performing process development and GMP manufacturing of bioconjugate for over 20 years, since the early 2000s. During this time, hundreds of projects has been successfully completed. With speed and flexibility, we can partner with clients from the earliest stages. That includes proof of concept, scale up, early to late stage generation of clinical materials, and recently, commercial launch. This slide depicts bioconjugation toolbox. At GBI, a wide range of bioconjugates has been developed, and it encompasses a wide range of targeting moieties. This includes mAbs and other mAb-like related molecules, recombinant proteins, pretty much any biomolecule recombinant protein we can handle, and also vaccines and carbohydrate type vaccines. GBI has different experience with a wide range of linkers that includes cleavable, non-cleavable linkers, chelators, and also in some cases, there's a direct conjugation whereby we can conjugate the linker and payload directly to the protein. A wide range of chemistries have been employed at GBI. That includes cysteine-type chemistry, maleimidal to form a thioether linkage. There's NHS ester, there's NCS, oxidative deamidation, and even click chemistry. A wide range of payload includes oligos, dyes, peptides, radionuclides, vaccine, pegylated, and the like. Over the years, GBI has developed conjugation strategies to develop the drug antibody ratio. That includes site-directed conjugation, typically involves reduction of one of the two disulfides at the hinge, say for an IgG1, for example. And in that case, we can actually add two payloads onto the antibody. There's also random conjugation, which gives you a Gaussian distribution of the drug to the antibody composition. And of course, there's also direct conjugation that I mentioned earlier, whereby there's no linker, the antibody is directly linked onto the payload. Applications for these conjugates include therapeutics, diagnostics, and theranostics. Next slide, please. Based on our deep experience of bioconjugates, a wide range of molecules has been successfully used. We can take a client project from earlier stages, proof of concept, all the way through to commercial launch. A wide range of these molecules include monoclonal antibodies, recombinant proteins, polymers, and peptides. Typical bioconjugate includes photoimmune conjugates, utilization of dyes, radioimmune conjugates, theranostic conjugates, pegylated, vaccine, and a wide range of other payloads. As part of our conjugation process development and manufacturing strategy, GBI has accumulated a wide range of equipment that allows us to precisely control the conjugation process. One example is the Advanced Synthesis Mettler-Toledo system. This is a workstation that allows us to control certain parameters from the earlier stage, small scale, all the way through to large scale manufacturing for commercial manufacturing. Next slide, please. This is just a snapshot of the very recent types of conjugates GBI has been working on, and these are ongoing. And it gives you some sense about the experience that GBI has accumulated over the last 20 years. And these projects, right now, they're either in the early stage proof of concept process development stage, and some of them are in the late stage. In fact, there is one now we just completed, the commercial launch. It's a complex bioconjugate. Next slide, please. At this stage, I would like to introduce Dr. Hung-Hee Lee. He's the senior scientist for bioconjugation process development. Over to you, Dr. Lee.
Thank you, Mukhtar. So antibody oligonucleotide conjugate, or AOCs, consist of antibodies and oligonucleotides. These oligonucleotides are small interfering RNAs, microRNAs, and antisense oligonucleotide. The field of AOCs started as a diagnostic tool. However, over past 10 years, AOCs have emerged as a novel therapeutic agent. Before using AOC as therapeutics, oligonucleotides alone have been used to treat genetic disorders. However, there are some obstacles. For example, unmodified oligonucleotides are unstable in serum and non-specifically accumulate in the cells. In addition, oligonucleotides have poor cell permeabilities. These issues have been improved using lipid-based delivery vehicles, such as liposomes or lipid nanoparticles. So far, 4 oligonucleotide drugs have been approved for the market. Interestingly, the delivery of all approved oligonucleotide-based drugs have primarily been targeted to the liver. This lack of tissue selectivity has been eventually resolved using antibodies as the targeted delivery vehicles. Because antibodies have high affinity and specificity for target antigens, so antibodies can selectively deliver oligonucleotide to targeted cells. Next speaker will be Dr. Natalia Bourguignon. She is MSET USB manager.
Thank you, Dr. Lee. Next slide, please. So currently, AOCs are generated mostly by 3 methods. The first one is the ionic interaction-based conjugation that involves the binding between the positively charged proamines linked to antibody and the negatively charged siRNAs to form an antibody siRNA conjugates. The second one is the avidin-biotin affinity-based conjugation. That is supported by the strong interaction between a biotin-labeled oligonucleotide and avidin-linked antibody. The third one is the direct chemical conjugation, and this is a method where an oligonucleotide is directly covalent bonded to an antibody molecule without the use of an intermediate linker. Essentially, attaching the 2 components through a chemical reaction between reactive groups on each molecule. This is similar to methods used for generating antibody-drug conjugates and other bioconjugates. Among the covalent binding, we can find first the random conjugation that occurs when an antibody with available amine groups will be randomly linked to an oligonucleotide chain. The other covalent binding is the site-directed conjugation. This involves the reduction of one of the disulfide bridges of the antibody via non-thiol reducing agent. The result is the formation of the 2 thiol reactive groups, and this is followed by coupling with the malamide linker oligonucleotide conjugate to afford the antibody-linked oligonucleotide product. The third one between the covalent binding is the direct conjugation, where an antibody will be directly coupled to an oligonucleotide without using an additional linker molecule by utilizing a carbohydrate EDC NAH ester chemistry to activate the carboxylic acid group on the oligonucleotide, allowing it to react the amine groups on the antibody, forming an established amide bond between the 2 molecules. Next slide, please. Due to the structural characteristics of the AOCs, some manufacturing steps as well as analytical methods for their characterization need to be modified or developed to address the production for clinical trials. Some considerations for the synthesis of AOCs are, for example, the scaling up, because involves scale up of amount of hazardous solvents used in the oligo-based drug manufacturing. Also, the analytical and purification methods will be developed specifically for AOC, because an oligonucleotide is larger than a typical drug linker and can have a molecular weight greater than 10 kilodalton. having a large influence on the physical and chemical properties of the conjugated antibody. Also, the analysis and characterization need to be adapted based on that the oligonucleotides are negative charged with over 20 negative charges. They are water soluble and will dominate the overall charge profile of the AOC. As part of the characterization, is needed to determine the oligonucleotide antibody ratio. This is a parameter used to describe the ratio of oligonucleotides to the antibody. Need to be determined by mass spectrometry, fluorescence, or PCR amplification. In addition, safety and facilities are important. Manufacturer must provide safe transport of the solvents to the facility and guarantee safe on-site storage. In addition, also, cold storage needs to be available to keep products and intermediate compounds. For a stable final formulation buffer, the utilization of appropriate excipients and/or additive may be needed to preserve the AOC stability and functionality. Next slide, please. GBI proposed an approach to AOC process development and GMP manufacturing by using site-directed conjugation. A project starts with the assessment of the technology and clinical manufacturing. This assessment involves the tech transfer and data by the client. A typical end-to-end project usually involves antibody production beginning from the research cell bank. This run comprises a process development, master cell bank creation, and GMP manufacturing of the antibody, and the development of antibody reduction process. This process generally takes about 8 months using GBI platforms. An alternative approach for the client is to provide a commercially available biosimilar for the antibody. The melamine linker oligonucleotide will be provided by a partner, and the next stage is the PD and GMP run for the AOC conjugation that involves synthesis, feasibility, conformance run, that is the PD run at scale to learn about the process and evaluate the scale. Also, the IRS is created and the assays are qualified before the GMP run. After the GMP run will be executed, and this set of steps involve the 4 to 5 months. The resultant AOC drug intermediate, drug substance, and drug product will be characterized to determine the OAR. Drug substance, IRS, is created and released, and after formulation is filled aseptically to afford the drug product. This takes about 4 to 5 months. The total end-to-end timeline proposed by GBI is about 16 to 18 months, considering that some of the activities may occur in parallel. GBI, as a flexible CDMO, can also partially execute the project, depending on the client needs. Next slide, please.
Thank you, Natalia. I'll just do a quick summary review of what we've learned today. We know that AOCs are an emerging field. They combine the specific binding of antibodies with the vast structural and functional properties of oligonucleotides. They're applicable to a variety of applications such as imaging detection, investorial class of generic biomolecules to use as therapeutic agents. This evolution includes addressing major challenges in AOC's manufacturing, and with over 20 years experience and knowledge of bioconjugates, GBI is an end-to-end CDMO uniquely qualified to provide solutions for AOC manufacturing at many stages, including up to commercial launch. Thank you for your attention, and we will go to a couple of questions that are commonly asked. First question, are AOCs stable? Natalia, will you answer this question, please?
Yes, of course. So AOC is a new class of bioconjugates that are highly complex and high-value products with many components associated with it. So to that end, each AOC stability has to be treated on a case-by-case basis and empirical development. A number of factors must be considered during the development of the stability program. This includes the size and type of the oligonucleotide being used, the type of monoclonal antibody or mAb-like proteins. We also need to consider the utilization of the appropriate additives and excipients, such as lipids, which is part of the entire composite of the AOC. Of course, GBI will be happy to further discuss your PD manufacturing and product stability needs.
Thank you, Natalia. Our second question is, what are the scale-up challenges?
Scaling up the manufacturing of AOCs involves several challenges due to the complexity of both the biomolecular components, that are the antibodies, oligonucleotides, lipids, and excipients, and their conjugation process. Some considerations are conjugation reaction scale-up. It's important to establish process control such as maintain the precise stoichiometric ratios, reaction conditions, mixing speed, temp, and reduce heterogeneity of the product. Also, we can mention purification challenges during the scaling up, since the conjugate product often requires complex purification to separate unconjugated antibodies, free oligonucleotides, and partially conjugate intermediates. In addition, conjugates require specialized analytical methods to assess purity, conjugation efficiency, and stability. AOC are high-value products, so process economics such as yield and cost consideration are important too. Finally, the regulatory and GMP compliance considerations for the scaling up are critical in order to meet the appropriate product quality, such as safety, identity, potency, and purity.
And thank you again for that answer, Natalia. Here are some bioconjugation references that can be found on our website via these links. We at GBI thank you for your attention and time watching this webinar. Please contact us if you have any needs in oligonucleotides or antibody conjugates or other bioconjugates. Thank you again.